History and nature. Snake. Snakes are elongated, limbless, carnivorous reptiles of the suborder serpents, /Esarntis/. Like all other squamates, snakes are ectothermic, amniote vertebrates covered in overlapping scales. Many species of snakes have skulls with several more joints than their lizard ancestors, enabling them to swallow prey much larger than their heads, cranial kinesis. To accommodate their narrow bodies, snakes' paired organs, such as kidneys, appear one in front of the other instead of side by side, and most have only one functional lung. Some species retain a pelvic girdle with a pair of vestigial claws on either side of the cloaca. Lizards have independently evolved elongate bodies without limbs or with greatly reduced limbs at least 25 times via convergent evolution, leading to many lineages of legless lizards. These resemble snakes, but several common groups of legless lizards have eyelids and external ears, which snakes lack, although this rule is not universal. See Amphisbaenia, Dibamidae, and Pygopodidae. Living snakes are found on every continent except Antarctica, and on most smaller land masses. Exceptions include some large islands, such as Ireland, Iceland, Greenland, the Hawaiian archipelago, and the islands of New Zealand, as well as many small islands of the Atlantic and Central Pacific Oceans. Additionally, sea snakes are widespread throughout the Indian and Pacific Oceans. Around 30 families are currently recognized, comprising about 520 genera and about 3,900 species. They range in size from the tiny, 10.4 cm long, 4.1 in, Barbados thread snake to the reticulated python of 6.95 meters, 22.8 feet, in length. The fossil species Titanoboa ceruginoensis was 12.8 meters, 42 feet, long. Snakes are thought to have evolved from either burrowing or aquatic lizards, perhaps during the Jurassic period, with the earliest known fossils dating to between 143 and 167 ma ago. The diversity of modern snakes appeared during the Paleocene epoch, c. 66 to 56 ma ago, after the Cretaceous-Paleogene extinction event. The oldest preserved descriptions of snakes can be found in the Brooklyn Papyrus. Most species of snake are non-venomous, and those that have venom use it primarily to kill and subdue prey rather than for self-defense. Some possess venom that is potent enough to cause painful injury or death to humans. Non-venomous snakes either swallow prey alive or kill by constriction. Etymology. The English word snake comes from Old English snaka, itself from Proto-Germanic *snaken, cf. Germanic schnakering snake, Swedish snodgrass snake, from Proto-Indo-European root *s, nego to crawl to creep, which also gave sneak as well as Sanskrit naga snake. The word ousted adder, as adder went on to narrow in meaning, though in Old English nedra was the general word for snake. The other term, serpent, is from French, ultimately from Indo-European *serp to creep, which also gave ancient Greek ropaiomega, herpo, I crawl. Evolution. A phylogenetic overview of modern snakes. Scolicophidia, Leptotyphlopidae, Anomalopitidae, Typhlopidae, Alethinophidia, Amorophidia, Anilius, Tropidophiidae, Afrophidia, Europeltoidea, Europeltidae, Anomochelus, Cylindrophis, Macrostomata, Pythonoidea, Pythonidae, Xenopeltis, Loxocemus, Xenophidia, Acrocordidae, Xenodermidae, Periidae, Viperidae, Homolopsidae, Colubridae, Cyclocoridae, Buhoma, Illepidae, Pseudospididae, Prosymnidae, Samophytiidae, Atractospididae, Pseudoxerophiidae, Lamprophidae, Buidea, Boidae, Ericinae, Calibaria, Ungeliophyni, Sanzinia, Candoia. Note, the tree only indicates relationships, not evolutionary branching times. The fossil record of snakes is relatively poor because snake skeletons are typically small and fragile, making fossilization uncommon. Fossils readily identifiable as snakes, though often retaining hind limbs, first appear in the fossil record during the Cretaceous period. The earliest known true snake fossils, members of the crown group serpents, come from the marine Cymoliophides, the oldest of which is the late Cretaceous, Cenomanian age, Haesiophis terrasanctus, dated to between 112 and 94 million years old. Based on comparative anatomy, there is consensus that snakes descended from lizards. Pythons and boas, primitive groups among modern snakes, have vestigial hind limbs, tiny, clawed digits known as anal spurs, which are used to grasp during mating. The families Leptotyphlopody and Typhlopody also possess remnants of the pelvic girdle, appearing as horny projections when visible. Front limbs are non-existent in all known snakes. This is caused by the evolution of their Hox genes, controlling limb morphogenesis. The axial skeleton of the snake's common ancestor, like most other tetrapods, had regional specializations consisting of cervical, neck, thoracic, chest, lumbar, lower back, sacral, pelvic, and caudal, tail, vertebrae. Early in snake evolution, the Hox gene expression in the axial skeleton responsible for the development of the thorax became dominant. As a result, the vertebrae anterior to the hind limb buds, when present, all have the same thoracic-like identity, except from the atlas, axis, and 1 to 3 neck vertebrae. In other words, most of a snake's skeleton is an extremely extended thorax. Ribs are found exclusively on the thoracic vertebrae. Neck, lumbar, and pelvic vertebrae are very reduced in number, only 2 to 10 lumbar and pelvic vertebrae are present, while only a short tail remains of the caudal vertebrae. However, the tail is still long enough to be of important use in many species, and is modified in some aquatic and tree-dwelling species. Many modern snake groups originated during the Paleocene, alongside the adaptive radiation of mammals following the extinction of non-avian dinosaurs. The expansion of grasslands in North America also led to an explosive radiation among snakes. Previously, snakes were a minor component of the North American fauna, but during the Miocene, the number of species and their prevalence increased dramatically with the first appearances of vipers and elapids in North America and the significant diversification of colubridae, including the origin of many modern genera such as Nerodia, Lampropeltis, Pituophis, and Pantherophis. Fossils. There is fossil evidence to suggest that snakes may have evolved from burrowing lizards during the Cretaceous period. An early fossil snake relative, Najash rheonogrina, was a two-legged burrowing animal with a sacrum and was fully terrestrial. One extant analog of these putative ancestors is the earless monitor Lanthanotus of Borneo, though it also is semi-aquatic. Subterranean species evolved bodies streamlined for burrowing, and eventually lost their limbs. According to this hypothesis, features such as the transparent, fused eyelids, brill, and loss of external ears evolved to cope with fossorial difficulties, such as scratched corneas and dirt in the ears. Some primitive snakes are known to have possessed hind limbs, but their pelvic bones lacked a direct connection to the vertebrae. These include fossil species like Haseophis, Pachyrachis, and Eupotophis, which are slightly older than Najash. This hypothesis was strengthened in 2015 by the discovery of a 113-million-year-old fossil of a four-legged snake in Brazil that has been named Tetrapodophis amplectus. It has many snake-like features, is adapted for burrowing, and its stomach indicates that it was preying on other animals. It is currently uncertain if Tetrapodophis is a snake or another species, in the squamate order, as a snake-like body has independently evolved at least 26 times. Tetrapodophis does not have distinctive snake features in its spine and skull. A study in 2021 places the animal in a group of extinct marine lizards from the Cretaceous period known as Dolichosaurs and not directly related to snakes. An alternative hypothesis, based on morphology, suggests the ancestors of snakes were related to mosasaurs, extinct aquatic reptiles from the Cretaceous, forming the clade Pythonomorpha. According to this hypothesis, the fused, transparent eyelids of snakes are thought to have evolved to combat marine conditions, corneal water loss through osmosis, and the external ears were lost through disuse in an aquatic environment. This ultimately led to an animal similar to today's sea snakes. In the late Cretaceous, snakes recolonized land and continued to diversify into today's snakes. Fossilized snake remains are known from early late Cretaceous marine sediments, which is consistent with this hypothesis, particularly so, as they are older than the terrestrial Najash rheonogrina. Similar skull structure, reduced or absent limbs, and other anatomical features found in both mosasaurs and snakes lead to a positive cladistical correlation, although some of these features are shared with varanids. Citation needed. Genetic studies in recent years have indicated snakes are not as closely related to monitor lizards as was once believed, and therefore not to mosasaurs, the proposed ancestor in the aquatic scenario of their evolution. However, more evidence links mosasaurs to snakes than to varanids. Fragmented remains found from the Jurassic and early Cretaceous indicate deeper fossil records for these groups, which may potentially refute either hypothesis. Tetrapodophis, Greek meaning four-footed snake, is an extinct genus of lizard from the early Cretaceous, Aptian, aged Cratoformation of Brazil. It has an elongate snake-like body, with 4 limbs. Tetrapodophis has been considered by some authors to be one of the oldest members of Ophidia, snakes and their extinct relatives. However, this classification has been disputed by some other authors, who identify Tetrapodophis as a dolichosaurid much less closely related to snakes. Description: Hindlimbs. Tetrapodophis possesses small yet well-developed fore and hind limbs like a lizard and a long body similar to a snake, around 19.5 centimeters, 7.7 in length. Nevertheless, it shares many characteristics with modern snakes, including an elongate body, short tail, broad belly scales, a skull with a short snout and long brain case, curved jaws, and sharp-hooked teeth. BMMSBK2-2 contains bones of another animal in its gut, indicating that Tetrapodophis was carnivorous like most snakes, but it turns out to be a lizard. Other features such as short neural spines suggest that Tetrapodophis was adapted to burrowing, lending support to the hypothesis that snakes evolved in terrestrial environments. Another hypothesis posits that they evolved in aquatic environments. The high number of vertebrae, upwards of 150, in Tetrapodophis and snakes is not seen in other burrowing reptiles with elongate bodies and reduced or absent limbs, meaning that it is most likely not an adaptation for a serpentine form of movement. Dolichosaurids are thought to be phylogenetically close to snakes as members of Ophidiomorpha, which explains some of the morphological similarities. Discovery and controversy. The type species, Tetrapodophis amplectus, was named in 2015 on the basis of a complete skeleton, BMMSBK2-2, preserved on a limestone slab in the Bürgermeister Müller Museum in Solnhofen, Germany, which was labeled as unknown fossil until its importance was recognized by paleontologist David Mardil, and illegally exported from Brazil, since the country's laws do not allow the removal of fossils from its territory, nor that studies on them be conducted without the participation of at least one Brazilian scientist. The specimen was later determined to come from the early Cretaceous Crato Formation in Ceará, Brazil. When asked why he didn't have a Brazilian scientist in the describing team, as required by Brazilian law, Mardil responded, "But what difference would it make? I mean, do you want me also to have a black person on the team for ethnicity reasons, and a cripple and a woman, and maybe a homosexual too just for a bit of all-round balance? If you invite people because they are Brazilian, then people will think that every Brazilian author on a scientific paper is there because he is Brazilian and not because he is a clever scientist." Mardil stated in 2020 that his comments were poorly worded. Classification. A phylogenetic analysis, published alongside the original 2015 description of Tetrapodophis, places it as a close relative of other lizards, but outside the crown group serpents, meaning that Tetrapodophis branched off before the most recent common ancestor of all living snakes. Below is a cladogram from that analysis. Eupodophis. Eupodophis is an extinct genus of snake from the late Cretaceous period. It has 2 small hind legs and is considered a transitional form between Cretaceous lizards and limbless snakes. The feature, described as vestigial, was most likely useless to Eupodophis. The type species Eupodophis descoensi was named in 2000 and resides now in the paleontology section of the MIM Museum in Beirut, Lebanon. The specific name is dedicated to the French naturalist Didier Descoings. The fossilized specimen from which the description of the type species was based was 85 centimeters, 33.5 long and is approximately 92 million years old. It was found in Cenomanian age limestone near the Al Namura village in Lebanon belonging to the Sannine Formation. Description: Eupotophus was a marine snake that lived in the Mediterranean Tethys Ocean. It had a laterally compressed body and short, paddle-like tail. The vertebrae and ribs of Eupotophus are pachyostotic, or thickened, as an adaptation to a marine lifestyle. The pelvic bones are small and weakly attached to each other. Tarsal bones are present but reduced in size and form. The metatarsals and phalanges of the foot are absent. The fossil skeleton of Eupotophus was analyzed using synchrotron X-rays at the European Synchrotron Radiation Facility in Grenoble, France. The researchers determined that the hind limb on one skeleton was 0.8 inches long, with an unmistakable fibula, tibia, and femur. One limb was visible on the surface of the fossil while the other was hidden within the limestone. The scans were compared with similar ones taken of the limbs of extant lizards, including the Gila monster, green iguana, and several species of monitor lizard. The hind limb of Eupotophus. While they are very small in comparison to limbed reptiles, the hind limbs of Eupotophus possess much of the same anatomy as modern lizards. This suggests that the bones of Eupotophus became reduced in size through a change in the rate of bone growth, not major anatomical changes. The lack of thickening at either end of the limb bones suggests that growth had stopped occurring in the limbs at one point in the animal's lifetime. While the vertebrae and ribs of Eupotophus are pachyostotic and osteosclerotic, meaning that the outer and inner parts of the bone are compact, the limb bones remain light. This lightness is also seen in the bones of terrestrial lizards, suggesting that the limbs had not been part of the overall adaptation of the skeleton for an aquatic lifestyle. Paleobiology. The loss of limbs in Eupotophus may have been the result of changes in Hox genes, genes that specify the development-specific regions of the body. Because Hox genes are involved in determining specific features of the axial skeleton, the loss of limbs would also result in the loss of cervical, tail, vertebrae that are near them. This loss is seen in Eupotophus and modern snakes, but not legless lizards, which may be far less common because some other factor besides Hox genes were involved in the loss of their limbs. The loss of digits on the hind limbs may be explained by a low number of cells in the limb bud during embryonic development. The loss of forelimbs and reduction of hind limbs in Eupotophus was likely an adaptation for swimming. While living snakes usually employ undulatory movement for moving over land, sinuous movements are also an effective means of moving through water. Large, well-developed limbs increase drag on swimming animals, so the limbs of Eupotophus and other early snakes may have become vestigial to save energy and make movement more efficient. Genetic basis of snake evolution. Main article: Limb development. Both fossils and phylogenetic studies demonstrate that snakes evolved from lizards, hence the question became which genetic changes led to limb loss in the snake ancestor. Limb loss is actually very common in extant reptiles and has happened dozens of times within skinks, anguids, and other lizards. In 2016, 2 studies reported that limb loss in snakes is associated with DNA mutations in the zone of polarizing activity regulatory sequence, ZRS, a regulatory region of the sonic hedgehog gene which is critically required for limb development. More advanced snakes have no remnants of limbs, but basal snakes such as pythons and boas do have traces of highly reduced, vestigial hind limbs. Python embryos even have fully developed hind limb buds, but their later development is stopped by the DNA mutations in the ZRS. Distribution. Approximate world distribution of snakes. There are about 3,900 species of snakes, ranging as far northward as the Arctic Circle in Scandinavia and southward through Australia. Snakes can be found on every continent except Antarctica, as well as in the sea, and as high as 16,000 feet, 4,900 meters, in the Himalayan mountains of Asia. There are numerous islands from which snakes are absent, such as Ireland, Iceland, and New Zealand, although New Zealand's northern waters are infrequently visited by the yellow-bellied sea snake and the banded sea kite. Taxonomy. See also, List of snake genera. All modern snakes are grouped within the suborder Serpents in Linnaean taxonomy, part of the order Squamata, though their precise placement within squamates remains controversial. The two inforders of serpents are Elathynophidia and Scolicophidia. This separation is based on morphological characteristics and mitochondrial DNA sequence similarity. Elathynophidia is sometimes split into Henophidia and Cynophidia, with the latter consisting of colubroid snakes, colubrids, vipers, elapids, hydrophiids, and atractaspids, and acrocordids, while the other Elathynophidian families comprise Henophidia, 40, while not extant today. The Madtsoidae, a family of giant, primitive, python-like snakes, was around until 50,000 years ago in Australia, represented by genera such as 1 ambi. There are numerous debates in the systematics within the group. For instance, many sources classify Boidae and Pythonidae as one family, while some keep the Elapidae and Hydrophydae, sea snakes, separate for practical reasons despite their extremely close relation. Recent molecular studies support the monopoly of the clades of modern snakes, Scolicophidians, Typhlopids plus Anomallepidans, Elathynophidians, Coralethynophidians, Europeltids, Cylindrophus, Anomochelys, Europeltans, Macrostomatans, Buids, Boids, Pythonids, and Cynophidians. Families. Infroder Elathynophidia, 25 families. Family, 5. Taxon author, 5. Genera, 5. Species, 5. Common name geographic range. Acrocordidae Bonaparte, 1831-13 Wart snakes Western India and Sri Lanka through tropical Southeast Asia to the Philippines, south through the Indonesian/Malaysian island group to Timor, east through New Guinea to the northern coast of Australia to Musso Island, the Bismarck Archipelago and Guadalcanal Island in the Solomon Islands. Aniliidae Stejniger, 1907-11 False coral snake tropical South America. Anomachilidae Kundal, Wallach, 1993-13 Dwarf pipe snakes West Malaysia and on the Indonesian island of Sumatra. Atractaspididae Gunther, 1858-1272 burrowing asps Africa and the Middle East. Boidae Gray, 1825-1461 boas northern, central, and South America, the Caribbean, southeastern Europe and Asia Minor, northern, central and East Africa, Madagascar and Reunion Island, the Arabian Peninsula, central and southwestern Asia, India and Sri Lanka, the Moluccas and New Guinea through to Melanesia and Samoa. Baleariidae Hofstetter, 1946-22 split-jaw snakes Mauritius. Colubridae Opal, 1811-58, 5, 2055, 5, typical snakes widespread on all continents, except Antarctica. Cyclocoridae Waynell and Brown, 2017 5-8 Cyclocorids the Philippines. Cylindrophydae Fitzinger, 1843-114 Asian pipe snakes Sri Lanka east through Myanmar, Thailand, Cambodia, Vietnam and the Malay archipelago to as far east as Aru Islands off the southwestern coast of New Guinea. Also found in southern China, Fujian, Hong Kong and on Hainan Island, and in Laos. Elapidae Boy, 1827-55 389 elapids on land, worldwide in tropical and subtropical regions, except in Europe. Sea snakes occur in the Indian Ocean and the Pacific, 43, Homolopsidae Bonaparte, 1845-2853 Homolopsids southeastern Asia and northern Australia. Lamprophidae Fitzinger, 1843-1689 Lamprophiids, formerly included Atracuspididae, Samophydiidae, and several other families, Africa, including the Seychelles. Loxosemidae Cope, 1861-11 Mexican burrowing snake along the Pacific versant from Mexico south to Costa Rica. Periidae Romer, 1956-320 snail-eating snakes Southeast Asia and islands on the Sunda Shelf, Sumatra, Borneo, Java, and their surrounding smaller islands. Procinidae Kelly, Barker, Villett and Broadley, 2009-116 shovel-snout snakes Sub-Saharan Africa. Samophydiidae Bourgeois, 1968-855 Samophiids Africa, including Madagascar, Asia, and southern Europe. Pseudospididae Cope, 1893-34 Pseudospidids mostly Sub-Saharan Africa, 2 species in Southeast Asia. Pseudoxyrophiidae Dowling, 1975-2289 Pseudoxyrophiids mostly Madagascar and the Comoros, 5 species in Sub-Saharan Africa, 1 in Socotra. Pythonidae Fitzinger, 1826-840 pythons sub-Saharan Africa, India, Myanmar, southern China, Southeast Asia and from the Philippines southeast through Indonesia to New Guinea and Australia. Tropidophyidae brongersma, 1951-234 dwarf boas West Indies, also Panama and northwestern South America, as well as in northwestern and southeastern Brazil. Europeltidae Muller, 1832-855 shield-tailed snakes southern India and Sri Lanka. Viperidae Oppel, 1811-35 341 vipers the Americas, Africa, and Eurasia east to Wallace's Line. Xenodermidae Cope, 1906-18 dragon and odd-scaled snakes southern and southeastern Asia, and islands on the Sunda Shelf, Sumatra, Borneo, Java, and their surrounding smaller islands. Xenopeltidae Bonaparte, 1845-12 sunbeam snakes Southeast Asia from the Andaman and Nicobar Islands, east through Myanmar to southern China, Thailand, Laos, Cambodia, Vietnam, the Malay Peninsula and the East Indies to Sulawesi, as well as the Philippines. Xenophidiidae Wallach and Gunther, 1998-12 spine-jawed snakes Borneo and peninsular Malaysia. Inferter Scolicophidia 5 families, family, 5, taxon author, 5, genera, 5, species, 5, common name geographic range. Anomalepide Taylor, 1939-418 primitive blind snakes from southern Central America to northwestern South America. Disjunct populations in northeastern and southeastern South America. Garipilidae Vidal, Wynne, Donnellan and Hedges 2010-218 Indo-Malayan blind snakes southern and southeastern Asia, including Sri Lanka, the Philippines, and New Guinea. Leptotyphlopidae Stegniger, 1892-13 139 slender blind snakes Africa, Western Asia from Turkey to northwestern India, on Socotra Island, from the southwestern United States south through Mexico and central to South America, though not in the high Andes. In Pacific South America they occur as far south as southern coastal Peru, and on the Atlantic side as far as Uruguay and Argentina. In the Caribbean they are found on the Bahamas, Hispaniola, and the Lesser Antilles. Typhlopidae Merem, 1820-18 266 typical blind snakes most tropical and many subtropical regions around the world, particularly in Africa, Madagascar, Asia, islands in the Pacific, tropical America, and in southeastern Europe. Xenotyphlopidae Vidal, Vences, Branch and Hedges 2010-11 round-nosed blind snake northern Madagascar. Legless lizards. Main article, legless lizard. While snakes are limbless reptiles, evolved from, and grouped with, lizards, there are many other species of lizards that have lost their limbs independently but which superficially look similar to snakes. These include the slowworm and glass snake. Other serpentine tetrapods that are unrelated to snakes include Sicilian, amphibians, amphisbaenians, near-lizard squamates, and the extinct istopods, amphibians. Biology. An adult Barbados thread snake, Leptotyphlops carli, on an American $quarter. Size. The now extinct Titanoboa ceruginoensis was 12.8 meters, 42 feet in length. By comparison, the largest extant snakes are the reticulated python, measuring about 6.95 meters, 22.8 feet long, and the green anaconda, which measures about 5.21 meters, 17.1 feet long and is considered the heaviest snake on Earth at 97.5 kilograms, 215 pounds. At the other end of the scale, the smallest extant snake is Leptotyphlops carli, with a length of about 10.4 centimeters, 4.1 in. Most snakes are fairly small animals, approximately 1 meter, 3.3 feet in length. Perception. Thermographic image of a snake eating a mouse. Pit vipers, pythons, and some boas have infrared-sensitive receptors in deep grooves on the snout, allowing them to see the radiated heat of warm-blooded prey. In pit vipers, the grooves are located between the nostril and the eye in a large pit on each side of the head. Other infrared-sensitive snakes have multiple, smaller labial pits lining the upper lip, just below the nostrils. A snake tracks its prey using smell, collecting airborne particles with its forked tongue, then passing them to the vomeronasal organ or Jacobson's organ in the mouth for examination. The fork in the tongue provides a sort of directional sense of smell and taste simultaneously. The snake's tongue is constantly in motion, sampling particles from the air, ground, and water, analyzing the chemicals found, and determining the presence of prey or predators in the local environment. In water-dwelling snakes, such as the anaconda, the tongue functions efficiently underwater. The underside of a snake is very sensitive to vibration, allowing the snake to detect approaching animals by sensing faint vibrations in the ground. Despite the lack of outer ears, they are also able to detect airborne sounds. Snake vision varies greatly between species. Some have keen eyesight and others are only able to distinguish light from dark, but the important trend is that a snake's visual perception is adequate enough to track movements. Generally, vision is best in tree-dwelling snakes and weakest in burrowing snakes. Some have binocular vision, where both eyes are capable of focusing on the same point, an example of this being the Asian vine snake. Most snakes focus by moving the lens back and forth in relation to the retina. Diurnal snakes have round pupils and many nocturnal snakes have slit pupils. Most species possess 3 visual pigments and are probably able to see 2 primary colors in daylight. It has been concluded that the last common ancestors of all snakes had UV-sensitive vision, but most snakes that depend on their eyesight to hunt in daylight have evolved lenses that act like sunglasses for filtering out the UV light, which probably also sharpens their vision by improving the contrast. Skin. Main article, Snake scale. The skin of a snake is covered in scales. Contrary to the popular notion of snakes being slimy, because of possible confusion of snakes with worms, snake skin has a smooth, dry texture. Most snakes use specialized belly scales to travel, allowing them to grip surfaces. The body scales may be smooth, keeled, or granular. The eyelids of a snake are transparent spectacle scales, also known as brill, which remain permanently closed. The shedding of scales is called ecdysis, or in normal usage, molting or sloughing. Snakes shed the complete outer layer of skin in one piece. Snake scales are not discrete, but extensions of the epidermis, hence they are not shed separately but as a complete outer layer during each molt, akin to a sock being turned inside out. Snakes have a wide diversity of skin coloration patterns which are often related to behavior, such as the tendency to have to flee from predators. Snakes that are at a high risk of predation tend to be plain, or have longitudinal stripes, providing few reference points to predators, thus allowing the snake to escape without being noticed. Plain snakes usually adopt active hunting strategies, as their pattern allows them to send little information to prey about motion. Blotched snakes usually use ambush-based strategies, likely because it helps them blend into an environment with irregularly shaped objects, like sticks or rocks. Spotted patterning can similarly help snakes to blend into their environment. The shape and number of scales on the head, back, and belly are often characteristic and used for taxonomic purposes. Scales are named mainly according to their positions on the body. In advanced, Cynophidian snakes, the broad belly scales and rows of dorsal scales correspond to the vertebrae, allowing these to be counted without the need for dissection. Molting. A common water snake shedding its skin. Molting, or ecdysis, serves a number of purposes. It allows old, worn skin to be replaced and it can remove parasites such as mites and ticks that live in the skin. It's also been observed in snakes that molting can be synced to mating cycles. Shedding skin can release pheromones and revitalize color and patterns of the skin to increase attraction of mates. Renewal of the skin by molting supposedly allows growth in some animals such as insects, but this has been disputed in the case of snakes. Molting occurs periodically throughout the life of a snake. Before each molt, the snake stops eating and often hides or moves to a safe place. Just before shedding, the skin becomes dull and dry-looking and the snake's eyes turn cloudy or blue-colored. The inner surface of the old skin liquefies, causing it to separate from the new skin beneath it. After a few days, the eyes become clear and the snake crawls out of its old skin, which splits close to the snake's mouth. The snake rubs its body against rough surfaces to aid in the shedding of its old skin. In many cases, the the cast skin peels backward over the body from head to tail in one piece, like pulling a sock off inside out, revealing a new, larger, brighter layer of skin which has formed underneath. A young snake that is still growing may shed its skin up to 4 times a year, but an older snake may shed only once or twice a year. The discarded skin carries a perfect imprint of the scale pattern, so it is usually possible to identify the snake from the cast skin if it is reasonably intact. This periodic renewal has led to the snake being a symbol of healing in medicine, as pictured in the rod of Asclepius. Scale counts can sometimes be used to identify the sex of a snake when the species is not distinctly sexually dimorphic. A probe is fully inserted into the cloaca, marked at the point where it stops, then removed and measured against the subcaudal scales. The scalation count determines whether the snake is a male or female, as the hemipenes of a male will probe to a different depth, usually longer, than the cloaca of a female. Clarification needed. Skeleton. The skeletons of snakes are radically different from those of most other reptiles, as compared with the turtle here, for example, consisting almost entirely of an extended ribcage. The skeleton of most snakes consists solely of the skull, hyoid, vertebral column, and ribs, though Henophidian snakes retain vestiges of the pelvis and rear limbs. The skull consists of a solid and complete neurocranium, to which many of the other bones are only loosely attached, particularly the highly mobile jaw bones, which facilitate manipulation and ingestion of large prey items. The left and right sides of the lower jaw are joined only by a flexible ligament at the anterior tips, allowing them to separate widely, and the posterior end of the lower jaw bones articulate with a quadrate bone, allowing further mobility. The mandible and quadrate bones can pick up ground-borne vibrations, because the sides of the lower jaw can move independently of one another. A snake resting its jaw on a surface has sensitive stereo-auditory perception, used for detecting the position of prey. The jaw quadrate stapes pathway is capable of detecting vibrations on the angstrom scale, despite the absence of an outer ear and the lack of an impedance matching mechanism, provided by the ossicles in other vertebrates, for receiving vibrations from the air. The hyoid is a small bone located posterior and ventral to the skull, in the neck region, which serves as an attachment for the muscles of the snake's tongue, as it does in all other tetrapods. The vertebral column consists of between 200 and 400 vertebrae, or sometimes more. The body vertebrae each have 2 ribs articulating with them. The tail vertebrae are comparatively few in number, often less than 20% of the total, and lack ribs. The vertebrae have projections that allow for strong muscle attachment, enabling locomotion without limbs. Caudal autotomy, self-amputation of the tail, a feature found in some lizards, is absent in most snakes. In the rare cases where it does exist in snakes, caudal autotomy is intervertebral, meaning the separation of adjacent vertebrae, unlike that in lizards, which is intravertebral, i.e. the break happens along a predefined fracture plane present on a vertebra. In some snakes, most notably boas and pythons, there are vestiges of the hind limbs in the form of a pair of pelvic spurs. These small, claw-like protrusions on each side of the cloaca are the external portion of the vestigial hind limb skeleton, which includes the remains of an ilium and femur. Snakes are polyphyodonts with teeth that are continuously replaced. Internal organs. Anatomy of a snake. File info: esophagus, trachea, tracheal lungs, rudimentary left lung, right lung, heart, liver, stomach, air sac, gallbladder, pancreas, spleen, intestine, testicles, kidneys. Snakes and other non-archosaur, crocodilians, dinosaurs plus birds and allies, reptiles have a 3-chambered heart that controls the circulatory system via the left and right atrium, and 1 ventricle. Internally, the ventricle is divided into 3 interconnected cavities: the cavum arteriosum, the cavum pulmonale, and the cavum venosum. The cavum venosum receives deoxygenated blood from the right atrium and the cavum arteriosum receives oxygenated blood from the left atrium. Located beneath the cavum venosum is the cavum pulmonale, which pumps blood to the pulmonary trunk. The snake's heart is encased in a sac, called the pericardium, located at the bifurcation of the bronchi. The heart is able to move around, owing to the lack of a diaphragm. This adjustment protects the heart from potential damage when large ingested prey is passed through the esophagus. The spleen is attached to the gallbladder and pancreas and filters the blood. The thymus, located in fatty tissue above the heart, is responsible for the generation of immune cells in the blood. The cardiovascular system of snakes is unique for the presence of a renal portal system in which the blood from the snake's tail passes through the kidneys before returning to the heart. The vestigial left lung is often small or sometimes even absent, as snakes' tubular bodies require all of their organs to be long and thin. In the majority of species, only one lung is functional. This lung contains a vascularized anterior portion and a posterior portion that does not function in gas exchange. This saccular lung is used for hydrostatic purposes to adjust buoyancy in some aquatic snakes, and its function remains unknown in terrestrial species. Many organs that are paired, such as kidneys or reproductive organs, are staggered within the body, one located ahead of the other. Snakes have no lymph nodes. Venom. Snake venom, venomous snake, and bite. Innocuous milk snakes are often mistaken for coral snakes whose venom is deadly to humans. Cobras, vipers, and closely related species use venom to immobilize, injure, or kill their prey. The venom is modified saliva, delivered through fangs. The fangs of advanced venomous snakes like viperids and elapids are hollow, allowing venom to be injected more effectively, and the fangs of rear-fanged snakes such as the boomslang simply have a groove on the posterior edge to channel venom into the wound. Snake venoms are often prey-specific, and their role in self-defense is secondary. Venom, like all salivary secretions, is a predigestant that initiates the breakdown of food into soluble compounds, facilitating proper digestion. Even non-venomous snake bites, like any animal bite, cause tissue damage. Certain birds, mammals, and other snakes, such as king snakes, that prey on venomous snakes have developed resistance and even immunity to certain venoms. Venomous snakes include 3 families of snakes, and do not constitute a formal taxonomic classification group. The colloquial term poisonous snake is generally an incorrect label for snakes. A poison is inhaled or ingested, whereas venom produced by snakes is injected into its victim via fangs. There are, however, 2 exceptions. Rhabdophis sequesters toxins from the toads it eats, then secretes them from nuchal glands to ward off predators, and a small unusual population of garter snakes in the U.S. state of Oregon retains enough toxins in their livers from ingested newts to be effectively poisonous to small local predators, such as crows and foxes. Snake venoms are complex mixtures of proteins, and are stored in venom glands at the back of the head. In all venomous snakes, these glands open through ducts into grooved or hollow teeth in the upper jaw. The proteins can potentially be a mix of neurotoxins, which attack the nervous system, hemotoxins, which attack the circulatory system, cytotoxins, which attack the cells directly, bungarotoxins, related to neurotoxins, but also directly affect muscle tissue, and many other toxins that affect the body in different ways. Almost all snake venom contains hyaluronidase, an enzyme that ensures rapid diffusion of the venom. Venomous snakes that use hemotoxins usually have fangs in the front of their mouths, making it easier for them to inject the venom into their victims. Some snakes that use neurotoxins, such as the mangrove snake, have fangs in the back of their mouths, with the fangs curled backwards. This makes it difficult both for the snake to use its venom and for scientists to milk them. Elapids, however, such as cobras and krites, are proteroglyphous. They possess hollow fangs that cannot be erected toward the front of their mouths, and cannot stab like a viper. They must actually bite the victim. It has been suggested that all snakes may be venomous to a certain degree, with harmless snakes having weak venom and no fangs. According to this theory, most snakes that are labeled non-venomous would be considered harmless because they either lack a venom delivery method or are incapable of delivering enough to endanger a human. The theory postulates that snakes may have evolved from a common lizard ancestor that was venomous, and also that venomous lizards like the Gila monster, beaded lizard, monitor lizards, and the now extinct mosasaurs may have derived from this same common ancestor. They share this venom clade with various other saurian species. Venomous snakes are classified in two taxonomic families: elapids, cobras including king cobras, krites, mambas, Australian copperheads, sea snakes, and coral snakes. Viperids, vipers, rattlesnakes, copperheads/cottonmouths, and bushmasters. There is a third family containing the Opistoglyphus, rear-fanged, snakes, as well as the majority of other snake species: colubrids, boomslangs, tree snakes, vine snakes, cat snakes, although not all colubrids are venomous. Reproduction. See also, sexual selection in scaled reptiles. Although a wide range of reproductive modes are used by snakes, all employ internal fertilization. This is accomplished by means of paired, forked hemipenes, which are stored, inverted, in the male's tail. The hemipenes are often grooved, hooked, or spined, designed to grip the walls of the female's cloaca. The clitoris of the female snake consists of two structures located between the cloaca and the scent glands. Most species of snakes lay eggs which they abandon shortly after laying. However, a few species, such as the king cobra, construct nests and stay in the vicinity of the hatchlings after incubation. Most pythons coil around their egg clutches and remain with them until they hatch. A female python will not leave the eggs, except to occasionally bask in the sun or drink water. She will even shiver to generate heat to incubate the eggs. Some species of snake are ovoviviparous and retain the eggs within their bodies until they are almost ready to hatch. Several species of snake, such as the boa constrictor and green anaconda, are fully viviparous, nourishing their young through a placenta as well as a yolk sac. This is highly unusual among reptiles, and normally found in requiem sharks or placental mammals. Retention of eggs and live birth are most often associated with colder environments. The garter snake has been studied for sexual selection. Sexual selection in snakes is demonstrated by the 3,000 species that each use different tactics in acquiring mates. Ritual combat between males for the females they want to mate with includes topping, a behavior exhibited by most viperids in which one male will twist around the vertically elevated forebody of its opponent and force it downward. It is common for neck biting to occur while the snakes are entwined. Facultative parthenogenesis. Parthenogenesis is a natural form of reproduction in which growth and development of embryos occur without fertilization. Agkistrodon contortrix, copperhead, and Agkistrodon piscivorus, cottonmouth, can reproduce by facultative parthenogenesis, meaning that they are capable of switching from a sexual mode of reproduction to an asexual mode. 83. The most likely type of parthenogenesis to occur is automixis with terminal fusion, a process in which two terminal products from the same meiosis fuse to form a diploid zygote. This process leads to genome-wide homozygosity, expression of deleterious recessive alleles, and often to developmental abnormalities. Both captive-born and wild-born copperheads and cottonmouths appear to be capable of this form of parthenogenesis. Reproduction in squamate reptiles is almost exclusively sexual. Males ordinarily have a ZZ pair of sex-determining chromosomes, and females a ZW pair. However, the Colombian rainbow boa, Epicrates morus, can also reproduce by facultative parthenogenesis, resulting in production of WW female progeny. The WW females are likely produced by terminal automixis. Embryonic development. Mouse embryo 12-day post-fertilization side by side with corn snake embryo 2 days post-ovopositioning. Snake embryonic development initially follows similar steps as any vertebrate embryo. The snake embryo begins as a zygote, undergoes rapid cell division, forms a germinal disc, also called a blastodisc, then undergoes gastrulation, neurulation, and organogenesis. Cell division and proliferation continues until an early snake embryo develops and the typical body shape of a snake can be observed. Multiple features differentiate the embryologic development of snakes from other vertebrates, two significant factors being the elongation of the body and the lack of limb development. Diagram illustrating differential somite size due to difference in somatogenesis clock oscillation. The elongation in snake body is accompanied by a significant increase in vertebra count. Mice have 60 vertebrae, whereas snakes may have over 300. This increase in vertebrae is due to an increase in somites during embryogenesis, leading to an increased number of vertebrae which develop. Somites are formed at the presemitic mesoderm due to a set of oscillatory genes that direct the semitogenesis clock. The snake semitogenesis clock operates at a frequency 4 times that of a mouse, after correction for developmental time, creating more somites, and therefore creating more vertebrae. This difference in clock speed is believed to be caused by differences in lunatic fringe gene expression, a gene involved in the semitogenesis clock. There is ample literature focusing on the limb development/lack of development in snake embryos and the gene expression associated with the different stages. In basal snakes, such as the python, embryos in early development exhibit a hind limb bud that develops with some cartilage and a cartilaginous pelvic element, however this degenerates before hatching. This presence of vestigial development suggests that some snakes are still undergoing hind limb reduction before they are eliminated. There is no evidence in basal snakes of forelimb rudiments and no examples of snake forelimb bud initiation in embryo, so little is known regarding the loss of this trait. Recent studies suggests that hind limb reduction could be due to mutations in enhancers for the SSH gene. However, other studies suggested that mutations within the HOX genes or their enhancers could contribute to snake limblessness. Since multiple studies have found evidence suggesting different genes played a role in the loss of limbs in snakes, it is likely that multiple gene mutations had an additive effect leading to limb loss in snakes. Behavior: winter dormancy. Snake coiled on a stick in Oklahoma. It was brumating in a large pile of wood chips, found by this landscaper after he bulldozed the pile in late autumn 2018. In regions where winters are too cold for snakes to tolerate while remaining active, local species will enter a period of brumation. Unlike hibernation, in which the dormant mammals are actually asleep, brumating reptiles are awake but inactive. Individual snakes may brumate in burrows, under rock piles, or inside fallen trees, or large numbers of snakes may clump together in hibernacula. Feeding and diet. African egg-eating snake eating an egg. All snakes are strictly carnivorous, preying on small animals including lizards, frogs, other snakes, small mammals, birds, eggs, fish, snails, worms, and insects. Snakes cannot bite or tear their food to pieces, so must swallow their prey whole. The eating habits of a snake are largely influenced by body size. Smaller snakes eat smaller prey. Juvenile pythons might start out feeding on lizards or mice and graduate to small deer or antelope as an adult, for example. The snake's jaw is a complex structure. Contrary to the popular belief that snakes can dislocate their jaws, they have an extremely flexible lower jaw, the two halves of which are not rigidly attached, and numerous other joints in the skull, which allow the snake to open its mouth wide enough to swallow prey whole, even if it is larger in diameter than the snake itself. 91, for example. The African egg-eating snake has flexible jaws adapted for eating eggs much larger than the diameter of its head. This snake has no teeth, but does have bony protrusions on the inside edge of its spine, which it uses to break the shell when eating eggs. Carpet python constricting and consuming a chicken. The majority of snakes eat a variety of prey animals, but there is some specialization in certain species. King cobras and the Australian bandy consume other snakes. Species of the family Pariidae have more teeth on the right side of their mouths than on the left, as they mostly prey on snails and the shells usually spiral clockwise. Some snakes have a venomous bite, which they use to kill their prey before eating it. Other snakes kill their prey by constriction, while some swallow their prey when it is still alive. Dolichophus jugularis, preying on a Sheltopusik. After eating, snakes become dormant to allow the process of digestion to take place. This is an intense activity, especially after consumption of large prey. In species that feed only sporadically, the entire intestine enters a reduced state between meals to conserve energy. The digestive system is then upregulated to full capacity within 48 hours of prey consumption. Being ectothermic, cold-blooded, the surrounding temperature plays an important role in the digestion process. The ideal temperature for snakes to digest food is 30 degrees Celsius 86 degrees Fahrenheit. There is a huge amount of metabolic energy involved in a snake's digestion. For example, the surface body temperature of the South American rattlesnake, Crotalus dorissus, increases by as much as 1.2 degrees Celsius (2.2 degrees Fahrenheit) during the digestive process. If a snake is disturbed after having eaten recently, it will often regurgitate its prey to be able to escape the perceived threat. When undisturbed, the digestive process is highly efficient. The snake's digestive enzymes dissolve and absorb everything but the prey's hair, or feathers, and claws, which are excreted along with waste. Hooding and spitting. Hooding, expansion of the neck area, is a visual deterrent, mostly seen in cobras, elapids, and is primarily controlled by rib muscles. Hooding can be accompanied by spitting venom towards the threatening object and producing a specialized sound, hissing. Studies on captive cobras showed that 13 to 22% of the body length is raised during hooding. Locomotion. The lack of limbs does not impede the movement of snakes. They have developed several different modes of locomotion to deal with particular environments. Unlike the gaits of limbed animals, which form a continuum, each mode of snake locomotion is discrete and distinct from the others. Transitions between modes are abrupt. Lateral undulation. Main article, undulatory locomotion. Crawling prints of a snake. Lateral undulation is the sole mode of aquatic locomotion, and the most common mode of terrestrial locomotion. In this mode, the body of the snake alternately flexes to the left and right, resulting in a series of rearward moving waves. While this movement appears rapid, snakes have rarely been documented moving faster than 2 body lengths per second, often much less. This mode of movement has the same net cost of transport, calories burned per meter moved, as running in lizards of the same mass. Terrestrial lateral undulation is the most common mode of terrestrial locomotion for most snake species. In this mode, the posteriorly moving waves push against contact points in the environment, such as rocks, twigs, irregularities in the soil, etc. Each of these environmental objects, in turn, generates a reaction force directed forward and towards the midline of the snake, resulting in forward thrust while the lateral components cancel out. The speed of this movement depends upon the density of push points in the environment, with a medium density of about 8, clarification needed, along the snake's length being ideal. The wave speed is precisely the same as the snake's speed, and as a result, every point on the snake's body follows the path of the point ahead of it, allowing snakes to move through very dense vegetation and small openings. When swimming, the waves become larger as they move down the snake's body, and the wave travels backwards faster than the snake moves forwards. Thrust is generated by pushing their body against the water, resulting in the observed slip. In spite of overall similarities, studies show that the pattern of muscle activation is different in aquatic versus terrestrial lateral undulation, which justifies calling them separate modes. All snakes can laterally undulate forward, with backward-moving waves, but only sea snakes have been observed reversing the motion, moving backwards with forward-moving waves. Sidewinding. Main article, sidewinding. A neonate sidewinder rattlesnake, Crotalus cerastes, sidewinding, most often employed by colubrids snakes, colubrids, elapids, and vipers, when the snake must move in an environment that lacks irregularities to push against, rendering lateral undulation impossible, such as a slick mud flat or a sand dune. Sidewinding is a modified form of lateral undulation in which all of the body segments oriented in one direction remain in contact with the ground, while the other segments are lifted up, resulting in a peculiar rolling motion. This mode of locomotion overcomes the slippery nature of sand or mud by pushing off with only static portions on the body, thereby minimizing slipping. The static nature of the contact points can be shown from the tracks of a sidewinding snake, which show each belly scale imprint, without any smearing. This mode of locomotion has very low caloric cost, less than 1/3 of the cost for a lizard to move the same distance. Contrary to popular belief, there is no evidence that sidewinding is associated with the sand being hot. Concertina. Main article, concertina movement. When push points are absent, but there is not enough space to use sidewinding because of lateral constraints, such as in tunnels, snakes rely on concertina locomotion. In this mode, the snake braces the posterior portion of its body against the tunnel wall while the front of the snake extends and straightens. The front portion then flexes and forms an anchor point, and the posterior is straightened and pulled forwards. This mode of locomotion is slow and very demanding, up to 7 times the cost of laterally undulating over the same distance. This high cost is due to the repeated stops and starts of portions of the body as well as the necessity of using active muscular effort to brace against the tunnel walls. Arboreal. Golden tree snake climbing a flower. The movement of snakes in arboreal habitats has only recently been studied. While on tree branches, snakes use several modes of locomotion depending on species and bark texture. In general, snakes will use a modified form of concertina locomotion on smooth branches, but will laterally undulate if contact points are available. 108. Snakes move faster on small branches and when contact points are present, in contrast to limbed animals, which do better on large branches with little clutter. Gliding snakes, Chrysopelia, of Southeast Asia launch themselves from branch tips, spreading their ribs and laterally undulating as they glide between trees. These snakes can perform a controlled glide for hundreds of feet depending upon launch altitude and can even turn in midair. Rectilinear. Main article, rectilinear locomotion. The slowest mode of snake locomotion is rectilinear locomotion, which is also the only one where the snake does not need to bend its body laterally, though it may do so when turning. In this mode, the belly scales are lifted and pulled forward before being placed down and the body pulled over them. Waves of movement and stasis pass posteriorly, resulting in a series of ripples in the skin. The ribs of the snake do not move in this mode of locomotion, and this method is most often used by large pythons, boas, and vipers when stalking prey across open ground, as the snake's movements are subtle and harder to detect by their prey in this manner. Interactions with humans. Most common symptoms of any kind of snake bite and venomation. Furthermore, there is vast variation in symptoms between bites from different types of snakes. Bite. Main article, snake bite. Vipirobarus, one fang in glove with a small venom stain, the other still in place. Snakes do not ordinarily prey on humans. Unless startled or injured, most snakes prefer to avoid contact and will not attack humans. With the exception of large constrictors, non-venomous snakes are not a threat to humans. The bite of a non-venomous snake is usually harmless. Their teeth are not adapted for tearing or inflicting a deep puncture wound, but rather grabbing and holding. Although the possibility of infection and tissue damage is present in the bite of a non-venomous snake, venomous snakes present far greater hazard to humans. The World Health Organization, WHO, lists snakebite under the other neglected conditions category. Documented deaths resulting from snake bites are uncommon. Non-fatal bites from venomous snakes may result in the need for amputation of a limb or part thereof. Of the roughly 725 species of venomous snakes worldwide, only 250 are able to kill a human with one bite. Australia averages only one fatal snake bite per year. In India, 250,000 snake bites are recorded in a single year, with as many as 50,000 recorded initial deaths. The WHO estimates that on the order of 100,000 people die each year as a result of snake bites, and around 3 times as many amputations and other permanent disabilities are caused by snake bites annually. The treatment for a snake bite is as variable as the bite itself. The most common and effective method is through antivenom, or antivenin, a serum made from the venom of the snake. Some antivenom is species-specific, monovalent, while some is made for use with multiple species in mind, polyvalent. In the United States, for example, all species of venomous snakes are pit vipers, with the exception of the coral snake. To produce antivenom, a mixture of the venoms of the different species of rattlesnakes, copperheads, and cottonmouths is injected into the body of a horse in ever-increasing dosages until the horse is immunized. Blood is then extracted from the immunized horse. The serum is separated and further purified and freeze-dried. It is reconstituted with sterile water and becomes antivenom. For this reason, people who are allergic to horses are more likely to have an allergic reaction to antivenom. Antivenom for the more dangerous species, such as mambas, taipans, and cobras, is made in a similar manner in South Africa, Australia, and India, although these antivenoms are species-specific. Snake charmers. Main article, snake charming. The Indian cobra is the most common subject of snake charmings. In some parts of the world, especially in India, snake charming is a roadside show performed by a charmer. In such a show, the snake charmer carries a basket containing a snake that he seemingly charms by playing tunes with his flute-like musical instrument, to which the snake responds. The snake is in fact responding to the movement of the flute, not the sound it makes, as snakes lack external ears, though they do have internal ears. The Wildlife Protection Act of 1972 in India technically prohibits snake charming on the grounds of reducing animal cruelty. Other types of snake charmers use a snake and mongoose show, where the two animals have a mock fight. However, this is not very common, as the animals may be seriously injured or killed. Snake charming as a profession is dying out in India because of competition from modern forms of entertainment and environment laws prescribing the practice. Many Indians have never seen snake charming, and it is becoming a folktale of the past. Trapping. The Iralas tribe of Andhra Pradesh and Tamil Nadu in India have been hunter-gatherers in the hot, dry plains forests, and have practiced the art of snake catching for generations. They have a vast knowledge of snakes in the field. They generally catch the snakes with the help of a simple stick. Earlier, the Iralas caught thousands of snakes for the snakeskin industry. After the complete ban of the snakeskin industry in India and protection of all snakes under the Indian Wildlife Protection Act 1972, they formed the Irala Snake Catchers Cooperative and switched to catching snakes for removal of venom, releasing them in the wild after 4 extractions. The venom so collected is used for producing life-saving antivenom, biomedical research, and for other medicinal products. The Iralas are also known to eat some of the snakes they catch and are very useful in rat extermination in the villages. Despite the existence of snake charmers, there have also been professional snake catchers or wranglers. Modern-day snake trapping involves a herpetologist using a long stick with a V-shaped end. Some television show hosts, like Bill Host, Austin Stevens, Steve Irwin, and Jeff Corwin, prefer to catch them using bare hands. Consumption. A sea leopard snake, supposedly in Hydraspo corti, occupies a place of honor among the live delicacies on display outside a Guangzhou restaurant. Although snakes are not commonly thought of as food, their consumption is acceptable in some cultures and may even be considered a delicacy. Snake soup is popular in Cantonese cuisine, consumed by locals in the autumn to warm their bodies. Western cultures document the consumption of snakes only under extreme circumstances of hunger, with the exception of cooked rattlesnake meat, which is commonly consumed in Texas and parts of the Midwestern United States. Snake meat in a Taipei restaurant. In Asian countries such as China, Taiwan, Thailand, Indonesia, Vietnam, and Cambodia, drinking the blood of a snake, particularly the cobra, is believed to increase sexual virility. When possible, the blood is drained while the cobra is still alive, and it is usually mixed with some form of liquor to improve the taste. The use of snakes in alcohol is accepted in some Asian countries. In such cases, 1 or more snakes are left to steep in a jar or container of liquor, as this is claimed to make the liquor stronger, as well as more expensive. One example of this is the habu snake, which is sometimes placed in the Okinawan liqueur habushu, also known as habu sake. Snake wine is an alcoholic beverage produced by infusing whole snakes in rice wine or grain alcohol. First recorded as being consumed in China during the Western Zhou dynasty, this drink is considered an important curative and is believed to reinvigorate a person according to traditional Chinese medicine. Pets. In the Western world, some snakes are kept as pets, especially docile species such as the ball python and corn snake. To meet the demand, a captive breeding industry has developed. Snakes bred in captivity are considered preferable to specimens caught in the wild and tend to make better pets. Compared with more traditional types of companion animal, snakes can be very low-maintenance pets. They require minimal space, as most common species do not exceed 5 feet (1.5 meters) in length, and can be fed relatively infrequently, usually once every 5 to 14 days. Certain snakes have a lifespan of more than 40 years if given proper care. Symbolism. Main article, Serpent, Symbolism. The reverse side of the throne of Pharaoh Tutankhamun with 4 golden uraeus cobra figures. Gold with lapis lazuli, Valley of the Kings, Thebes, 1347 to 37 BCE. Snakes composing a bronze Karakian from the mythical Longinus River in Sicily. Imperial Japan depicted as an evil snake in a World War II propaganda poster. The smoking snake, insignia of the Brazilian Expeditionary Force in World War II. A common adder in the coat of arms of the Kajarvi municipality. In ancient Mesopotamia, Nera, the messenger god of Istaron, was represented as a serpent on Kadurus, or boundary stones. Representations of 2 intertwined serpents are common in Sumerian art and Neo-Sumerian artwork and still appear sporadically on cylinder seals and amulets until as late as the 13th century BC. The horned viper, Cerastes cerastes, appears in Kassite and Neo-Assyrian Kadurus and is invoked in Assyrian texts as a magical, protective entity. A dragon-like creature with horns, the body and neck of a snake, the forelegs of a lion, and the hind legs of a bird appears in Mesopotamian art from the Akkadian period until the Hellenistic period, 323 BC, 31 BC. This creature, known in Akkadian as the Muu, meaning furious serpent, was used as a symbol for particular deities and also as a general protective emblem. It seems to have originally been the attendant of the underworld god Ninazu, but later became the attendant to the Hurrian storm god Tishpak, as well as, later, Ninazu's son Ninjashida, the Babylonian national god Marduk, the scribal god Nabu, and the Assyrian national god Ashur. In Egyptian history, the snake occupies a primary role with the Nile cobra adorning the crown of the pharaoh in ancient times. It was worshipped as one of the gods and was also used for sinister purposes, murder of an adversary and ritual suicide. Cleopatra. Citation needed. The Ouroboros was a well-known ancient Egyptian symbol of a serpent swallowing its own tail. The precursor to the Ouroboros was the many-faced, a serpent with 5 heads, who, according to the Amduat, the oldest surviving book of the afterlife, was said to coil around the corpse of the sun god Ra protectively. The earliest surviving depiction of a true Ouroboros comes from the gilded shrines in the tomb of Tutankhamun. In the early centuries AD, the Ouroboros was adopted as a symbol by Gnostic Christians, and chapter 136 of the Pistis Sophia, an early Gnostic text, describes a great dragon whose tail is in its mouth. In medieval alchemy, the Ouroboros became a typical Western dragon with wings, legs, and a tail. In the Bible, King Nahash of Ammon, whose name means snake, is depicted very negatively, as a particularly cruel and despicable enemy of the ancient Hebrews. Medusa, 1597, by the Italian artist Caravaggio. The ancient Greeks used the Gorgonian, a depiction of a hideous face with serpents for hair, as an apotropaic symbol to ward off evil. In a Greek myth described by Pseudo-Apollodorus in his Bibliotheca, Medusa was a gorgon with serpents for hair whose gaze turned all those who looked at her to stone and was slain by the hero Perseus. In the Roman poet Ovid's Metamorphoses, Medusa is said to have once been a beautiful priestess of Athena, whom Athena turned into a serpent-haired monster after she was raped by the god Poseidon in Athena's temple. In another myth referenced by the Boeotian poet Hesiod and described in detail by Pseudo-Apollodorus, the hero Heracles is said to have slain the Lernaean Hydra, a multiple-headed serpent which dwelt in the swamps of Lerna. The legendary account of the foundation of Thebes mentioned a monster snake guarding the spring from which the new settlement was to draw its water. In fighting and killing the snake, the companions of the founder Cadmus all perished, leading to the term Cadmean victory, i.e. a victory involving one's own ruin. Citation needed. Rod of Asclepius, in which the snake, through ecdysis, symbolizes healing. 3 medical symbols involving snakes that are still used today are bowl of Hygeia, symbolizing pharmacy, and the caduceus and rod of Asclepius, which are symbols denoting medicine in general. One of the etymologies proposed for the common female first name Linda is that it might derive from Old German Lindi or Linda, meaning a serpent. India is often called the land of snakes and is steeped in tradition regarding snakes. Snakes are worshipped as gods even today, with many women pouring milk on snake pits, despite snakes' aversion for milk. The cobra is seen on the neck of Shiva, and Vishnu is depicted often as sleeping on a seven-headed snake or within the coils of a serpent. There are also several temples in India solely for cobras, sometimes called Nagraj, king of snakes, and it is believed that snakes are symbols of fertility. There is a Hindu festival called Nag Panchami each year, on which day snakes are venerated and prayed to. See also Naga, citation needed. In India there is another mythology about snakes. Commonly known in Hindi as Ichchadhari snakes. Such snakes can take the form of any living creature, but prefer human form. These mythical snakes possess a valuable gem called mani, which is more brilliant than diamond. There are many stories in India about greedy people trying to possess this gem and ending up getting killed, citation needed. The snake is one of the 12 celestial animals of Chinese zodiac, in the Chinese calendar. Many ancient Peruvian cultures worshipped nature. They emphasized animals and often depicted snakes in their art. Religion. Main article, Snake worship. Snakes are used in Hinduism as a part of ritual worship. In the annual Nag Panchami festival, participants worship either live cobras or images of Nagas. Lord Shiva is depicted in most images with a snake coiled around his neck. Puranic literature includes various stories associated with snakes. For example, Shisha is said to hold all the planets of the universe on his hoods and to constantly sing the glories of Vishnu from all his mouths. Other notable snakes in Hinduism are Vasuki, Takshaka, Karkataka, and Pingla. The term Naga is used to refer to entities that take the form of large snakes in Hinduism and Buddhism. Snakes have been widely revered in many cultures, such as in ancient Greece, where the serpent was seen as a healer. Asclepius carried a serpent wound around his wand, a symbol seen today on many ambulances. In Judaism, the snake of brass is also a symbol of healing, of one's life being saved from imminent death. In religious terms, the snake and jaguar were arguably the most important animals in ancient Mesoamerica. In states of ecstasy, lords dance a serpent dance. Great descending snakes adorn and support buildings from Chichen Itza to Tenochtitlan, and the Nahuatl word Codal, meaning serpent or twin, forms part of primary deities such as Mixcoatl, Quetzalcoatl, and Coatlicue. In the Maya and Aztec calendars, the 5th day of the week was known as Snake Day. In some parts of Christianity, the redemptive work of Jesus Christ is compared to saving one's life through beholding the Nehushtan, serpent of brass. Snake handlers use snakes as an integral part of church worship, to demonstrate their faith in divine protection. However, more commonly in Christianity, the serpent has been depicted as a representative of evil and sly plotting, as seen in the description in Genesis of a snake tempting Eve in the Garden of Eden. St. Patrick is purported to have expelled all snakes from Ireland while converting the country to Christianity in the 5th century, thus explaining the absence of snakes there. In Christianity and Judaism, the snake makes its infamous appearance in the first book of the Bible when a serpent appears before Adam and Eve and tempts them with the forbidden fruit from the tree of knowledge. The snake returns in the book of Exodus when Moses turns his staff into a snake as a sign of God's power, and later when he makes the Nehushtan, a bronze snake on a pole that when looked at cured the people of bites from the snakes that plagued them in the desert. The serpent makes its final appearance symbolizing Satan in the book of Revelation, and he laid hold on the dragon, the old serpent, which is the devil and Satan, and bound him for 1,000 years. In neo-paganism and Wicca, the snake is seen as a symbol of wisdom and knowledge. Additionally, snakes are sometimes associated with Hecate, the Greek goddess of witchcraft. Ballcourt marker from the post-classic site of Mixco Viejo in Guatemala. This sculpture depicts Kukulkan, jaws agape, with the head of a human warrior emerging from his maw. Medicine. Several compounds from snake venoms are being researched as potential treatments or preventatives for pain, cancers, arthritis, stroke, heart disease, hemophilia, and hypertension, and to control bleeding, e.g. during surgery. My name is Arya. Please like, share, subscribe and hit that notification bell so you can be the first to be notified whenever we post. You won't regret it.